Hello, we're continuing with life science and cell biology in a nutshell. So, currently, we're learning about the endoplasmic reticulum. Um, the difference we learned between the rough and the smooth endoplasmic reticulum is that smooth is um, more focused on the creation of lipids and steroids, while the rough is more um, just in charge of creating proteins and enzymes. Thank you for the ice cream, Ruben. So, as you can tell, there's an ice cream truck that is parked right next to me. So, we got some background music now. So, so, um, so the lipids that are created in the smoothie are, once again, they include various kinds of molecules. They include like steroids, lipids, which um, they include cholesterol, even vitamin D, etc. So such molecules. The smoothie R is contiguous or is continuous with the rough ER, but it doesn't have any ribosomes, and the ribosomes make proteins, so it doesn't have ribosomes. So um, the smoothie R has vesicles as well, which move away and carry molecules that are created, such as steroids and lipids, etc. In these vesicles too, the Golgi complex. So all the proteins from the rough ER and the lipids and steroid molecules from the smooth ER are carried to the Golgi body. Um, and so so they're like cargo basically that are the Golgi body, which we learned about earlier, is in charge of taking these different molecules and taking them to their different destinations within or outside the cell. So it is like it functions like a post office, which takes everything that and adds markers to them in order to um, have the molecules destined for specific locations, for specific functions. So, in summary, the various parts of the endomembrane system, so the cell membrane and all the different parts of the cell, the rough ER, the smooth ER work in conjunctions with the other parts of the endomembrane system to carry out the cell's functions and enable it to maintain homeostasis, which is a balanced environment with all the different molecules being at the levels and concentrations that they need to be to maintain a normal, healthy cell environment and healthy tissues. The structure of the endoplasmic reticulum enables it to fulfill its functions, and as such, both the rough and smooth ER have different functions and different appearances. Rough ER is dubbed rough because it is covered in ribosomes, which gives the organelle a bumpy, rough appearance. Ribosomes are structures responsible for making proteins. After the proteins are created, they are sent into the lumen, which is the hollow space within the tubules and sacs of the ER, then some proteins remain in the membrane of the ER, while others will be transferred where to other locations. So they can be other transferred to locations either within the cell or to the outer membrane and exported outside of the cell. So proteins inside the ER get folded. The process of protein folding means they're modified. An example of which is the fusion of carbohydrate side chains to the proteins. The modified proteins are then sent to a variety of destinations. Some are secreted from the cell via an organelle called a vesicle, um, which are found in the Golgi body. They're sent to carry out functions elsewhere in an animal's body, while others remain in the membrane of the ER or are moved to other organelles. So the rough ER is also responsible for creating phospholipids, which make up the membrane of cells. The amount of rough endoplasmic reticulum a cell has depends upon the function of the cell. Certain cells must secrete substantial amounts of proteins and enzymes for use in the body. As such, they have a higher amount of rough ER than other cells. Proteins that will not stay in the ER are packed into vesicles sent to the Golgi apparatus, which the Golgi is an organelle responsible for sending proteins to the right place in the cell or body by shuffling them via vesicles. All the molecules, proteins, lipids, etc., large macromolecules. It is often referred to as the post office of the cell, the Golgi body or complex. It does tagging, sorting, sending lipids and proteins to the correct places. So from the ER, and usually they are also modified further by the Golgi body before they are sent out. Other chains of molecules can be removed or added to the proteins, and phosphate groups can be used as a tag to indicate their correct destination. So also, lysosomes are another part of the endomembrane system. So this organelle recycles other old organelles, digesting them so that the molecules that make them up can be used or recycled to create new structures. Some of the vesicles that exit the Golgi body will head to the lysosome to release their cargo, or they're broken down in the lysosome. Lysosomes play a role in the immune system as well. They digest and degrade foreign or harmful particles from the outside of the cell. Macrophages are white blood cells which specialize in swarming over pathogens and engulfing them. This process is known as phagocytosis. The part of the pathogen that the cell engulfs is separated from the plasma membrane and encased in a structure referred to as a phagosome. It is then fused with the lysosome, where the digestive enzymes break down the pathogen, rendering it harmless. So the smooth and rough ER play important roles in eukaryotic cells. The ER is what kicks off the process